ACE Botany next episode. In this episode, we'll be discussing biology classification, fifth one. And what all things we'll be discussing in this episode? For that, I will share the screen, so it will be convenient for you. Basically, fungus, all the groups, after introduction, then 5 theory question, 5 NEET model question with the key. So I'll share it, so it will be convenient for you. You can find the notes also. I think screen came, so we can proceed. Biology classification, fifth one, fungus continued, mainly groups. So when we come to introduction, so in the last episode, fungus introduced and that aspect details discussed. Here we'll be discussing Phycomycete, Ascomycete, Basidio, and Deuteromycete. Each group, their habitat, habit, reproduction, economic aspect, and diseases. So after introduction, when we enter, we can find fungus. Basically 4 groups: Phycomycete, Ascomycete, Basidiomycete, and Deuteromycete. And what is the criterion for this grouping? Important question. Last episode also we discussed. Here also we can just explain it. 1, morphology of mycelium. 2nd, mode of spore formation and fruiting bodies. These are the basis. Important question for theory NEET. And first group we have to learn is Phycomycete. Lower fungus, we are starting algal fungi and can be broadly classified into 2, oomycete and zygomycetes. So when we start with the oomycetes, which is otherwise called oogamous fungus. See, this is comparatively more primitive fungus among these 4. Cenocytic mycelium, no cross wall, no septa. Cell wall, remember, fungal cell wall is normally chitin. But in oomycete, we can find fungal cell wall is cellulose, exception, and showing primitiveness. Cellulose normally will be finding or can be observed in green plants and algae. But in fungus, oomycete, we can also find cellulosic cell wall. And other chemicals also see. Asexual reproduction is by spores formed in sporangia. But in aquatic condition, we can find zoospores. Zoospores will be produced. And normally, it is biflagellate zoospores and having heteroconflagellation. One flagellum is smooth and other is thin cell type with the small hair-like structures, which is called mastigonemes. Sexual reproduction again in the starting stage, so planogametic fusion or gametangial contact, and oospore is produced. That is why the tempomycete, oospore is produced, undergo meiosis. And some famous diseases due to oomycete. This part is important. Connected with NCERT, I put some diseases and some previous questions came. All the disease is not easy. Basically, material is NCERT. White rust of crucifers, albugo candida, late blight of potato, Phytophthora infestans, Irish famine, 1845-1847, damping of seedlings, Pythium divarianum, seedlings will be affected, downy mildew of cereals, Sclerospora graminicola, green ear disease of Bajra, Sclerospora graminicola, downy mildew in pea mustard, Spinaconian, it is Peranospora parasitica. Some important diseases, that's all. Now we can proceed to next group, zygomycete conjugation fungus. It is a group of terrestrial fungus. Saprotrophic, very few parasitic, cell wall changed into chitin or fungal cellulose. Previous group, it was cellulose. Here it is chitin. So evolution started. Chitin or fungal cellulose. Then mycelium shows cenocytic nature. In this group, normally motile cells are absent. So non-motile mitospores, sporangiospores, they are formed at the tips of structures called sporangiophores. PHRE, already you know it is talk. Normal sexual reproduction, gametangial copulation or conjugation, zygomycete, which is otherwise called conjugation fungus, multinucleate gametes, that is otherwise called xenogametes produced. Now, zygospore is the resting diploid spore produced after sexual reproduction. Zygospore formation, again, zygomycete, conjugation fungus. Zygospore differs from oospore. So, how zygospore differs from oospore in that during its formation, a distinct large food-laden non-motile female gamete is not produced. So there is a variation or difference between zygospore and oospore. I think idea clear. Now, meiosis takes place within inverted comma zygospore. Why that inverted comma? But it will not produce new mycelium directly. Here it produces a new sporangium, which is called germ sporangium or zygosporangium. Germsporangium or zygosporangium forms meiospore, germ spores after meiosis. So starting with the zygospore. I think idea clear. Now one more exception is there, that is called azygospore. A in the beginning of zygospore, so opposite. Parthenogenetically produced zygospore. In certain cases, gametangia fail to fuse. So nature given a mechanism in the sense that gametangia will be surrounded by a thick wall. Then it is known as azygospore, typically rhizopus and mucor haploid in nature. I think up to that it is clear. Now some important specimens. One is Pilobolus crystallinus, which is coprophilous, squirting fungus dung mold. Rhizopus stolonifer, black bread mold. Mucormycido, again dung mold. Now three habit I drawn there, you can find them. First is Rhizopus, which is concerned with the soft rot or leak disease, and you can find labeled parts. From the top, sporangia you can find, inside it sporangiospores, and there is an area called columella, and there is a stalk called sporangiophore, a sexual reproductive structure in Rhizopus. Stolon will be there, Rhizolife will be there. Then comes second one, Pilobolus. Pilobolus is commonly called squirting fungus. There also you can find sporangia and vesicles, sporangiophore, etc. Then third is mucor, here label mucor, and mucormycido, also called cow dung. And mucor, that is causing infection to internal organs to humans. Then from mucor, an antibiotic called ramazine or amazine from mucor species has been extracted. And how this Rhizopus and mucor differs. Rhizopus we can find from the Rhizolife directly, sporangiophore arises. And 2-3 will be their minimum. Here we can find it is scattered and individually they arise. I think it is clear. Now we can proceed to next group, ascomycete, commonly called sac fungus. Mycelium is septate. Then yeast is an exception because it is unicellular. So all items, when we discuss exception, we can observe. The members are saprophytic, parasitic, decomposers, coprophilous. Since septa is there, septal pores will be there. Chitinous cell wall, motile structures do not occur in them. So advancement we can find there. There are different modes of asexual reproduction, but the most common is conidia, and conidia are formed on branched or unbranched hyphae called conidiophore. Here you can find Aspergillus, a mature conidiophore having sterigmata, chains of conidia, and the labeling I made based on color. So you can find vesicle, that is this part white in color, chains of conidia you can find yellow, then sterigmata, that is stalk, orange red, then conidia for this one, and bottom you can find root cell or foot cell, not root cell, foot cell. That is regarding Aspergillus, a mature conidiophore having sterigmata, chains of conidia. I think I did clear. With the color, you can easily identify. Now, conidia and conidia are formed on conidia, and conidia are formed on branched or unbranched hyphae called conidiophore, or means stalk. Ascogonium is the female sex organ. In sexual reproduction, plasmogamy by different methods. For karyogamy to take place, a delay will be there. So, in exceptional case, they're creating a transitional phase, N+N, which is otherwise called dikaryo phase, but it is only having short duration, short. There it differs from the next group. And there is a method called cross-year method, that is cross-year method from a dikaryotic cell, other cells also become dikaryotic by transferring the nucleus, and the phenomenon is called dikaryotization. Cross-year method in ascomycete. Further details not needed because NCERT not going up to that level. Even cross-year, I think, term is not there. Cross-year means hook-like structure. Now, as we proceed to next page, some of the dikaryotic cell will function as ascus mother cells, and this will convert the cell into ascae or ascus. Ascus is seen in ascomycete. It is just like a sporangial sac fungus. In ascus, karyogamy and meiosis takes place. 4 or 8 ascospores. You can find the diagram here. 4 or 8 ascospores haploid are formed. Ascospores are endogenous. Generally, half of the ascospores are one type and other half is another mating type, plus minus. Ascospores arrangement will be linearly in neurospora and irregularly in yeast. Now, ascocar is the assembly of ascae. In yeast, it is absent against single cell. Some members coming under ascomycete. Again, this is the difficult part to remember. Gradually, it will be in your mind by continuous reading. Exam point of view important also. Yeast, industrial uses. Penicillium antibiotic. Aspergillus commonly called weed of the laboratory. And Aspergillus is also called guinea pig of the plant kingdom. A poisonous chemical aflatoxin is connected with Aspergillus. Neurospora, drosophile of the plant kingdom for genetical experiments. Precise is called cup fungus. Claviceps is called ergot fungus. LST is produced from claviceps. Erysipe is connected with a disease powdery mildew of cereals. Morels and truffles, edible. Monasus purpurea is cholesterol lowering agent. Morchella, it is morels. So some are edible, some are economically important, some are antibiotic producing, and some are disease causing. Now we have to proceed to basidiomycete club fungus. This group of fungus is most commonly seen and most advanced fungus as their foodification is frequently large. Example is mushrooms. And they are best decomposers of food. They are able to digest cellulose. Okay. Beyond cellulose, they can also digest lignin. Normally, it is very difficult to digest lignin even for other fungus and bacteria. Motile structure is absent. Advancement already there. So primary mycelium and secondary mycelium present in this group. Primary mycelium is short-lived, opposite to the previous group, and it is monokaryotic. And primary mycelium monokaryotic stage multiplies by oidea, conical-like spores. While when we come to secondary mycelium is having longer lifespan and also dominant phase. And it is the N plus N dikaryote phase. Outgrowth can be seen on the sides of the septa, and that is through a item called clamp connection. One of the function of the clamp connection is for proper distribution of dikaryon during cell division. Just compare with the crosshair in ascomycete. And in basidiomycete, we can find doliopore septa. A barrel-shaped septa can be seen. Now, secondary mycelium, how it is able to perennate, that is in soil or wood by sclerosian rhizomorph. Secondary mycelium is able to perennate. Different types of spores are seen in dikaryote phase or connected with the dikaryote phase in connection with the multiplication. Teletospores, urodospores, acidiospores, chlamydospores. Interestingly, in the sexual reproduction, sex organs are not involved. Term is sexual reproduction, but sex organs not involved. Fusion takes place between basidiospores and other monokaryotic spores. One method. Other method is hyphae fuses with the spermaceum or spore. And even there can be fusion between hyphal cells of primary mycelia. Nuclear fusion, that is karyogyne and meiosis in basidia. A typical basidium produces. Diagram I'll show you, it will be easy for you. 4 basidiospores exogenous at the tips of certain outgrowth called sterigmata. Ascospore endogenous, basidiospore exogenous. And here you can find sterigmata. And some members coming under basidiomycete, agaricus mushroom edible, astilago causing spot disease, Puccinia rust fungus, black stem rust of wheat. Puccinia has got different types of spores, and life cycle is heteroaceous. Dot stool poisonous, bracket fungus or shell fungus, puffballs, armillaria or armillariella. Armillariella. That is a fungus, not armillaria. Armillariella. Hallucinogens, etc. Because a zoology organism is there. Here it is armillariella. Deuteromycete. Last group, fungi imperfecti. And deuteromycete group include those fungus where perfect stage. Perfect stage means sexual stage, not clearly identified. So that means they were only asexual or vegetative phase. If sexual stage is identified, that stage is shifted to respective group of fungus. I think idea clear. Now, main reproductive mode is only asexual. Asexual spore known as conidia. And mycelium is septate and branch. They can be saprophytes or parasite. But many members are decomposers of litter, means organic matter, so helps in mineral cycling. And some members under deuteromycete, Alternaria solani, hairy blight of potato, Colectotrichum, red rot of sugarcane, Trichoderma biological control agent, Cyclosporinae, Fusarium wilt of cotton. And Fusarium is a deuteromycetian fungus, fungi imperfecti. But there is another fungus called Gibberella fusarium, is the perfect stage of this Fusarium. And Gibberella fusarium is concerned with the plant hormone gibberellins. So Fusarium and Gibberella fusarium. Cercospora tica disease, Helminthosporium or rice, Bengal famine, and that is otherwise called brown lace spot of rice. So some important diseases are there in all groups. Exam point of view question. Then as usual, 5 theory question, 5 NEET model question with the key. So what all things we learned in this episode? So in this episode, that is bioclassification 5th episode, we are mainly discussing different fungal group, starting with introduction, then fungus classification, criteria, Then phycomycete, lower algal fungi, two groups, oomycete, oogamous fungus, and zygomycete. Then, what are the features of oomycete? That is cellulosic cell wall. Asexual reproduction is by spores. Aquatic conditions, zoospore produce. Biflagellate zoospores, heteroconflagellation. Then, we can find primitive type of sexual reproduction, oospore produced, undergo meiosis, and some important specimens coming under oomycete and diseases. Then zygomycete, conjugation fungus, cell wall, chemical is chitin, fungal cellulose, mycelium still cynocytic, motile cells are absent, non-motile mitospores, sporangiospores. Then normal sexual reproduction is gametophageal copulation or conjugation. Then zygomycete, conjugation fungus, multinuclear gametes, xenogametes, and we can also find zygospore differs from oospore in that during its formation, distinct large food-laden non-motile female gamete is not produced. Meiosis, inverted comma, zygospore, but it will not produce new mycelium directly. Here it produces neosporangium, j-sporangium, zygosporangium. There forms meiospore, j-spore after meiosis. A zygospore is parthenogenetically produced zygospore, Rhizopus muca. Then some members with a diagram, that is Rhizopus, then Pilobolus, muca, common terms, disease, etc. Then sac fungus, ascomycete, septate hyphae, yeast exception, unicella. Then septal pores will be there. Then chitinous cell wall, motile structures do not occur. Then most common method of asexual reproduction, conidia. Then here you can find Aspergillus mixture conidia for having steric chains of conidia. Asconium is the female sex organ. Plasmogamy by different methods. Karyogamete to take place, a delay will be there. In a transition phase called N+ and dikaryote phase, it is having short duration by cross-sear method. Then dikaryotic cell, other cells become dikaryotic. From a dikaryotic cell, other cells also become dikaryotic. By transferring nucleus, phenomenon is called dikaryotization. Then some of it will be converted into ascus mother cell. Ascae or ascus will be formed. Karyogamete meiosis takes place in ascus. 4 to 8 ascospores form endogenous. And we can find generally half of the ascospores are one type, other half is another type plus minus. Ascospores are linear in neurospora and irregular in yeast. Then in yeast, ascocarp is absent. Some members coming under ascomycete, then basidiomycete, club fungus, most commonly seen and most advanced. Best decomposers of wood, can even digest lignin. Motile structure is absent. Then primary mycelium is short-lived and monokaryotic. Monokaryotic stage multiplies by ovoidea and conidia-like spores. Then secondary mycelium is having longer lifespan, also dominant phase. Then dikaryote phase, there is an outgrowth called outgrowth like septum, that is clamp connection. One of the functions of the clamp connection is for proper distribution of dikaryon during cell division. Doliopore septa is saying barrel-shaped septa. Secondary mycelium perination, sclerotia, and rhizomorphs. Different types of spores can be seen. Then sex organs not involved in sexual reproduction. Other types of fusion takes place. Nuclear fusion, karyogamete and meiosis in basidia. A typical basidium producers. 4 basidiospores, exogenous at the tips, diagram you can find. There's a stock called stercomata, then members coming under basidiomycete, then came deuteromycete, fungi imperfective. That is, perfect stage is not seen. So only asexual vegetative type of reproduction, like conidia, mycelium septate branched, and many members are decomposers of litter. They help in mineral cycling. Some important members coming under deuteromycete. Then after that, 5 theory question, 5 NEET model question with the key. If you like this episode, subscribe all, like it, share it, comment on it. Meanwhile, short question I'm daily putting. So to get all this, subscribe the channel. So take care. Bye. We can meet later. Thank you.